In this video, we're talking about fibrinolysis and coagulation screening. We're discussing and understanding the mechanisms of blood clotting, but also lysis of that clot to repair normal blood flow. Fibrinolysis is a process of breaking down a blood clot by degrading fibrin into soluble fragments. It is primarily mediated by plasmin, which is activated by plasminogen from tissue plasminogen activator and urokinase. This process restores normal blood flow after vessel repair and prevents excessive clotting. Fibrinolysis is a regulated and limited process that breaks down fibrin in blood clots to restore blood flow. It is controlled to prevent excessive clot breakdown, which could lead to excessive bleeding. And if it's not adequately monitored, you could lead to excessive clotting. It's regulated between a specific balance between pro-antihemostatic mediators and antifibrinolytic mediators. When a clot needs to be broken down, plasminogen activates plasmin by tissue plasminogen activator and urokinase, which will degrade fibrin into those soluble fragments. Regulation occurs through inhibitors like plasminogen activator inhibitor, which blocks tissue plasminogen activator, and alpha-2 antiplasmin, which inhibits plasmin activity, ensuring clot breakdown only occurs when and where it is needed. The fibrinolytic system is a tightly regulated process responsible for breaking down fibrin clots after vessel repair to restore normal blood flow and maintain that hemostatic balance. It ensures that clots are removed once their purpose is served, while preventing excessive clot degradation that could lead to bleeding. The key components include plasminogen, which is an inactive precursor protein produced in the liver, which is converted to plasmin, the active enzyme responsible for degrading fibrin. Plasmin is that main enzyme that breaks down fibrin into those soluble fragments called fibrin degrading products or D-dimers. Plasmin also degrades fibrinogen, factor V, and factor VIII. Now there's activators of plasminogen that include tissue plasminogen activator, released by endothelial cells. It binds to fibrin and converts plasminogen to plasmin, promoting localized fibrinolysis. Urokinase plasminogen activator is found in plasma and tissues and converts plasminogen to plasma, but is not fibrin dependent. Now, inhibitors of fibrinolysis, plasminogen activated inhibitor inhibits tissue plasminogen activator and urokinase, preventing premature activation of plasminogen, and alpha-2 antiplasmin, which directly inhibits plasmin activity to prevent systemic clot breakdown. So the steps in the fibrinolytic process, when there's a clot formation, that fibrin clot is formed via the coag cascade to stop bleeding. Plasminogen activation, it binds to the fibrin clot from the endothelial cells and activates plasminogen, converting it to plasmin. Then we have fibrin degradation, where plasmin cleaves fibrin into fibrin degrading products, including D-dimers, which are used as markers for clot breakdown in clinical tests. Regulation prevents excessive plasminogen activation, while alpha-2 antiplasmin inactivates plasmin that escapes the clot site, ensuring localized fibrinolysis. So you see the fibrinolytic system has inhibitors and activators that work together that regulate the clotting breakdown process. Plasmin degradation of fibrin is a process in which plasmin, a serine protease, breaks down fibrinogen into smaller soluble fragments. This process is essential for controlling clot formation and promoting clot removal. Unlike fibrin, which forms an insoluble network in clots, fibrinogen is soluble, so its degradation does not directly dissolve clots, but it prevents their formation and occurs in a series of stages or steps. First, plasminogen, an inactivated precursor, is activated to plasmin by tissue plasminogen activator or urokinase, where we have the cleavage of fibrinogen. Plasmin cleaves the alpha, beta, and gamma chains of fibrinogen at specific sites, releasing soluble fragments known as fibrinogen degrading products. The formation of FDPs, so they have major fragments that include X, Y, D, and E. The fragment X is a full-length fibrinogen with some cleaved sites. Fragment Y is a smaller intermediate fragment, and fragments D and E are the final degradation products that are released into the bloodstream. D-dimer is not produced from fibrinogen, but from the cross-linked fibrin, which is specific for clot breakdown. Now, the end of degradation includes the smaller fragments D and E that are soluble and cleared from circulation by the liver and the kidneys. When we discuss the control of hemostasis, we have to ensure that the clot formation is localized to the site of injury, where we prevent excessive clotting or thrombosis, or excessive bleeding called hemorrhage. The process is tightly regulated through a balance of procoagulant, anticoagulant, and fibrinolytic mechanisms. We can think of this in three different ways. First, we can think of it as procoagulant control, meaning clot formation. This mechanism promotes clot formation at the injury site, first by platelet activation, so platelets adhere to exposed collagen and von Willebrand, releasing ADP and thromboxane A2 to recruit more platelets. Then the coagulation cascade occurs. The intrinsic, extrinsic, and common pathways are activated, leading to the formation of thrombin, which converts fibrinogen to fibrin, stabilizing the clot. Positive feedback, so thrombin amplifies its own production by activating factors 5, 8, and 11, which accelerates clot formation. Now, next is anticoagulant control or clot inhibition. This prevents excessive clot formation and keeps clotting localized to the site. So when we talk about antithrombin, it inhibits thrombin or factor 2 and other activated factors like 10A, 9A, 11A, 12A, and it's enhanced by heparin. Protein C and S, this is where thrombin binds to thrombomodulin on endothelial cells, activating protein C, and protein C along with S inactivates factor 5A and 8A, slowing down the coagulation process. Tissue factor pathway inhibitor inhibits 7A tissue factor complex in the extrinsic pathway, reducing factor 10 activation. Fibrinolytic control is really the clot breakdown. This is a mechanism where we break down the clot once the vessel is repaired. And plasmin degrades the fibrin into fibrin degradation products. Plasmin is activated by plasminogen by tissue plasminogen activator. And plasmin also has inhibitors. So we want to inhibit TPA and UPA, while alpha-2 antiplasmin directly inhibits plasmin, ensuring clot breakdown occurs at the injury site. And we really want to have local control where the endothelium plays a vital role in controlling hemostasis by promoting both procoagulant and anticoagulant factors. So we have positive, negative feedback mechanisms to control the procoagulant versus anticoagulant abilities. We have the endothelial vasculature helping with the blood flow or limiting the blood flow. Then the liver has to clear or remove any clotting factors or complexes to prevent excess clotting. And we have inhibitors in the fibrinolytic system to ensure that it's all maintained and cleared out after the fact. So in the last slide, we talked about positive and negative regulators that control hemostasis. Please look over this chart to understand the positive feedback mechanisms or functions. This is really procoagulant type, and what are the components within this to help promote clotting. Then the negative feedback is what can we do to promote excessive clotting and prevent actual balance and control. So look over this chart. Okay, we're going to talk about basic screening tests for bleeding and clotting scenarios, where for coag testing, the initial screening test for PT-PTT includes a light blue sodium citrated tube, which has to be full at a 9:1 ratio of specimen to anticoagulant. If underfilled, we can have falsely prolonged tests, and if it's overfilled, clotting can occur in the tube, which also falsely prolongs results. It's used for routine hemostatic testing on citrated or platelet-poor plasma. Prothrombin time, or PT, is a screening test used for inherited or acquired deficiencies in extrinsic pathway, or can be used to monitor anticoagulant therapy, where the translocation of tissue factor to the cell surface activates the coag cascade by formation of tissue factor 7A complex. How we do this in the lab: we add thromboplastin to a patient's citrated platelet-poor plasma, and we record the time of fibrin formation or clotting time in seconds. So the reference range for PT is 10 to 15 seconds. If we have a prolongation of that time, we could consider problems or deficiencies or inhibitors in factors 1, 2, 5, 7, and 10. We can also consider monitoring oral anticoagulants such as Coumadin or warfarin, or any types of proteins induced by vitamin K absence. Activated partial thromboplastin time, or APTT, is a screening test used for inherited or acquired deficiencies of proteins within the intrinsic pathway, or circulating inhibitors to that pathway. We can also use it to monitor heparin therapy. How we do it in the lab is we have the patient's citrated plasma, and we have an APTT reagent called actin, which is an optimal activation of contact factors in order to help initiate the intrinsic process. They're incubated at 37 degrees, which is the normal body temperature within a human. Then to initiate the clotting cascade, we add calcium, and the time to fibrin formation is recorded. The general reference range is 25 to 35 seconds, and if we see a prolongation, we might consider deficiencies in 1, 2, 5, 8, 9, 10, 11, and 12.